Welcome to Unit 5 Microbiology for CUET PG Life Sciences. Microbiology is the branch of biology that studies microscopic organisms such as bacteria, fungi, viruses, protozoa, and algae. These organisms may be tiny in size, but they have enormous biological importance. They influence human health, ecosystem functioning, agriculture productivity, and industrial production processes. Understanding microbiology helps us understand how life functions at its most fundamental level. From an experimental and exam point of view, this unit is highly integrated and concept based. It connects cellular structure, metabolism, reproduction, immunity, and ecological interactions into one framework. As we move through this lecture, focus on understanding mechanisms, how microbes grow, how they interact with hosts, and how humans use them. This conceptual clarity will help you answer analytical and application based CUET PG questions. This slide explains how microbiology is assisted in the CUET PG examination. The exam does not simply test memory of definitions, it tests whether you understand biological mechanisms and relationships. For example, instead of asking what an antibody is, the exam may ask how antibodies function during immune response or how they differ structurally from other proteins. Another important exam trend is comparison and functional interruption. You may be asked to define bacterial classification methods, stages of bacterial growth, or types of immunity. Therefore, while studying, always connect structure to function and cause to effect. If you understand biological processes step by step, solving MCQs become much easier. Microbial diversity refers to the wide variety of microorganisms present in nature. Microbes differ in cellular organization, metabolism, ecological role, and evolutionary origin. Some are prokaryotic, other eukaryotic, and some are acellular like viruses. They exist in nearly every environment: soil, water, air, extreme habitats, and even inside living organisms. This diversity is essential for maintaining ecological balance. Microbes perform crucial biological functions such as decomposition of organic matters, nutrient cycling, oxygen production, and nitrogen fixation. Some microbes form beneficial symbiotic relationships while others cause diseases. Understanding this diversity helps explain the complexity of life systems and is fundamental for microbial classification questions in the exam. Microorganisms are broadly categorized into major groups based on cellular structure and biological organization. Bacteria and archaea are prokaryotes, meaning they lack a membrane-bound nucleus and organelles. However, archaea differ from bacteria in genetic makeup, biochemical pathways, and membrane structure, making them evolutionary distinct. Eukaryotic microorganisms include fungi, protozoa, and algae, which have complex internal organization with membrane-bound organelles. Viruses are unique because they are not true cells. They are acellular infectious particles that depend entirely on host cells for replication. Understanding these major groups helps build the foundation for classification, structural biology, and comparative microbiology. Bacteria are classified using multiple criteria. that reflect their structural and physiological differences. One basic classification is morphology, the physical shape of bacterial cells. Cocci or spherical, bacilli or rod shape, spirilla or spiral, and vibrio or comma shaped. These shapes influence movement, surface attachment, and colony formation. Another important classification system is gram staining, which distinguishes bacteria based on cell wall composition. Bacteria are also classified according to oxygen requirement, nutrient mode, and temperature tolerance. These classification methods help microbiologists identify species, predict metabolic behavior, and understand environmental adaptations. Archaea are prokaryotic organisms but are fundamentally different from bacteria at the molecular and biochemical level. These membrane lipids are chemically distinct, providing exceptional stability in extreme environmental conditions. These cell walls also lack peptidoglycan, which is a major structural component of bacterial cell wall. Many archaea are extrophilous, meaning they thrive in extreme environments such as hot springs, salt lakes, and oxygen-free habitats. Methanogens produce methane as a metabolic byproduct and play an important role in global carbon cycling. These adaptations make archaea important for understanding evolutionary biology and environmental microbiology. Fungi are eukaryotic heterophytic organisms that obtain nutrients through absorption. Their cell walls contain chitin, a strong structural polymer that provides rigidity and protection. Unlike plants, fungi do not perform photosynthesis. Instead, they secrete enzymes that break down organic matter externally and absorb the resulting nutrients. Fungi may be unicellular, like yeast, or multicellular, like molds, composed of filamentous hyphae. They reproduce through spores, which may be produced sexually or asexually. Ecologically, fungi act as a major decomposer and nutrient recycler, and economically, they are important in food production, fermentation, and antibiotic synthesis. Bacteria reproduce primarily through asexual processes that allow rapid population increase. Because bacterial cells are structurally simple and lack complex mitotic machinery, they can divide quickly under favorable conditions. This rapid reproduction allows bacteria to colonize environments efficiently. Understanding bacterial reproduction is essential for multiple biological fields. In medicine, it explains how infections spread rapidly. In biotechnology, it allows mass cultivation of bacteria for industrial production. In ecology, it explains how microbial populations respond quickly to the environmental changes. Binary fission is the primary mode of bacterial reproduction. The process begins with replication of the bacterial chromosome, followed by cell elongation. The two DNA copies move to opposite ends of the cell, and a spectrum forms in the center, dividing the cell into two daughter cells. The resulting daughter cells are genetically identical to the parent cell because binary fission is efficient and requires minimal cellular machinery. Bacterial populations can grow exponentially in a short time. This rapid multiplication has important implications in infections, dynamics, and microbial ecology. When bacteria grow in a closed system, their population follows a characteristic growth pattern called the bacterial growth curve. The first phase is the lag phase, where cells adapt to the new environment and synthesize enzymes necessary for growth but not divide rapidly. The next phase is the log or exponential phase, where cells divide rapidly and population size increases dramatically. This is followed by the stationary phase, where nutrient depletion and waste accumulation slow growth. Finally, in the death phase, cells die faster than they reproduce due to unfavorable conditions. Even though bacteria reproduce asexually through binary fission, they are capable of exchanging genetic material through process collectively called horizontal gene transfer. This genetic recombination increases variation in bacterial changes such as antibiotic exposure or nutrient availability. Unlike sexual reproduction in eukaryotes, bacterial recombination does not involve gamete formation. There are three major mechanisms of genetic recombination in bacteria: transformation, transduction, and conjugation. Transformation involves uptake of free DNA from the environment. Transduction occurs when bacteriophages transfer DNA between bacterial cells. Conjugation requires direct cell-to-cell contact and involves transfer of plasmid DNA through a sex pilus. These mechanism plays major role in bacterial evaluation and antibiotic resistance. Antimicrobial agents are substances that kill or inhibit the growth of microorganisms. They are essential tool in the medicine for treatment infectious diseases and in industry for controlling microbial contamination. These agents may be naturally produced by microorganisms, chemically synthesized or chemisynthetic modifications of nature components. The effectiveness of antimicrobial agents depends on selectively toxicity, the ability to harm microbes without damaging host cell. They achieve this by targeting structures or metabolic pathways unique to microorganisms, such as cell wall synthesis, ribosomal protein synthesis, or nucleic acid treatment strategies. Antimicrobial agents are classified based on the type of organism they target. Antibiotics specifically act against bacteria, antifungal agents target fungal pathogens, antiviral drugs interfere with viral replication, and antiparasitic drugs control protozoan or helminths infection. Each characteristic or category is designed to interfere with biological processes unique to the group. Antibiotics function by interfering with essential biological processes in bacterial cells. One common mechanism is inhibition of cell wall synthesis, which weakens structural integrity and causes osmotic lysis. This mechanism is effectively because human cells lack cell walls, maintaining the drug selectively toxic to bacteria. Antimicrobial resistance occurs when microorganisms develop the ability to survive exposure to drugs that once killed them. This is an evolutionary process driven by natural selection. When antibiotics are used, susceptible bacteria die while resistant ones survive and multiply, leading to resistant population. Microorganisms plays an important role in industrial production because of their ability to carry out diverse biochemical reactions efficiently. Industrial microbiology uses microbes to synthesize useful products such as enzymes, organic acids, alcohols, antibiotics, and biofuels. Their rapid growth and metabolic flexibility make them ideal protection systems. Industries cultivate microbes under controlled conditions to optimize growth and product yield. Fermentation technology is commonly used where microbes convert substrates into valuable products. This application demonstrates how microbial metabolism can be harassed for large-scale economic benefit. Microbial fermentation is widely used in food and beverage industry. Yeast converts sugars into alcohol during brewing and baking. Lactic acid bacteria ferment milk to produce yogurt and cheese. These processes depend on microbial metabolic pathway that transform raw materials into conceptual products. Microbes are also used in pharmaceutical industries to produce antibiotics, vaccines, and therapeutic proteins. Industrial enzymes produced by microbes are used in detergents, textiles, and food processing. These applications highlight the economic and technological importance of bacteria. Specific microorganisms are selected for industrial process based on their metabolic capability. Yeast species such as Saccharomyces cerevisiae are widely used in ethanol fermentation. The efficient sugar metabolism makes them ideal for beavering on and biofuel production. Fungi like penicillium produce antibiotics such as penicillin, which revolutionize medicine. Bacteria like lactobacillus produce lactic acid in dairy fermentation. Microorganisms play a crucial role in agriculture by maintaining soil fertility and promoting plant growth. They participate in nutrient cycling, organic matter decomposition, and formation of beneficial symbiotic relationship with plant roots. These interactions enhance plant productivity. Microorganisms improve agriculture through several mechanisms. Nitrogen fixing bacteria converts atmospheric nitrogen into forms usable by plants, increasing soil fertility. Biofertilizers contain beneficial microbes that enhance nutrient availability and root growth. Biopesticides derived from microbes help control pests without harmful chemical residues. Plant growth promoting microbes also produce hormones that stimulate plant development. These microbial activities make agriculture more sustainable and productivity. Nitrogen fixation is the biological process by which atmospheric nitrogen gas is converted into ammonia, a form that plants can observe and use to synthesize proteins and nucleic acids. Although nitrogen is abundant in the atmosphere, most organisms cannot utilize it directly because molecular nitrogen is chemically inert. Nitrogen fixing microorganisms solve this problem by using specialized enzymes, primarily nitrogenase, to break the strong triple bond of nitrogen gas. The immune system protects the body by identifying and eliminating foreign substances known as antigens. When an antigen enters the body, it triggers an immune response that involves recognition, signaling, and elimination. This defense system relies on specialized molecules and cells that detect and neutralize pathogen efficiently. An antigen is any molecule capable of triggering an immune response in the body. Typically, antigens are foreign proteins or polysaccharides present on the surface of pathogen such as bacteria, virus, or fungi. The immune system recognizes specifically regions of the antigen called epitopes, which interact directly with the immune receptor. Antibodies, also known as immunoglobulins, are Y-shaped protein produced by plasma cells derived from B lymphocytes. Each antibody consists of two heavy chains and two light chains linked by disulfide bonds. The tips of the Y-shaped structure from variable regions that binds specifically to antigen epitopes. There are different classes of antibodies including IgG, IgM, IgA, IgE, and IgD, each with specialized function. Some neutralize pathogens directly, others activate complement, and some mediate allergic responses. Structural variation allows antibodies to perform diverse immune functions while maintaining specificity. The complement system consists of a group of plasma proteins that circulate in inactive form and become activated in a sequential cascade when triggered by pathogens. Once activated, complement proteins amplify immune responses and help eliminate invade microorganisms. Complement activation leads to several protective effects, including formation of a membrane attack complexes that lyse pathogens, enhancement of phagocytosis through optimization, and stimulation of inflammation. Complement pathway may be activated through classical or alternative mechanisms depending on how the immune system detects the pathogen. The immune system is broadly divided into innate immunity and adaptive immunity. These two components work together to defend the body against infection. Innate immunity is the body's first line of defense and is present from birth. It includes physical barriers such as skin and mucous membrane, as well as cellular defense like macrophages and neutrophils. These components act rapidly to prevent pathogen entry and spread. Adaptive immunity develops after exposure to specific antigens and provides highly targeted defense. It involves specialized lymphocytes, B cells, and T cells that recognize specific molecular structure on pathogen. This specifically allows precise elimination of invading organisms. Vaccines are biological preparations designed to stimulate the immune system without causing disease. They introduce harmless form or component of pathogen to train the immune system to recognize and respond to real infection in the future. Types of vaccines. Vaccines are classified based on how the antigen is prepared. Live attenuated vaccine contains weakened forms of pathogens that can replicate but do not cause disease in healthier individuals. Inactivated vaccines contain killed pathogen that cannot replicate but still stimulate immunity. Other types include subunit vaccines containing specific pathogen component, toxicoid vaccine using inactivated toxins, and modern mRNA vaccines that instruct cells to produce antigenic proteins. Each type differs in safety, immune response strength, and duration of protection. The principle of vaccination is based on the ability of the immune system to develop memory after exposure to an antigen. When the body encounters a pathogen for the first time, it takes time to generate an immune response. However, during this response, memory B cells and T cells are formed. These cells remain in the body and allow a much faster and stronger response in the same pathogen enter again. Plant viruses are infectious agents that specifically infect plant cells and disrupt normal plant growth and development. Most plant viruses contain RNA as a genetic material, although some may contain DNA. They lack independent metabolic machinery and rely entirely on host plant cells for replication. Plant viruses typically cause characteristic disease symptoms such as mosaic pattern on leaves, discoloration, leaf curling, and stunning growth. These symptoms result from disruption of chloroplast function and interference with Normal cellular mechanism. The pattern of symptoms often helps in identifying specific viral infection. One of the most studied plant viruses is tobacco mosaic virus, which infects tobacco and many other plant species. It produces characteristic mosaic patterns on leaves and reduces photosynthetic efficiency. It was the first virus ever discovered and remains an important model organism in the virology. Animal virus infect cells of animals and humans, often causing disease. Like all viruses, they consist of genetic material surrounded by a protein coat called a capsid. Some animal viruses also possess an outer lipid envelope derived from host cell membrane during viral release. A virus consists of two main structural components, genetic material and a protein capsid that protects the genome. The genetic material may be DNA or RNA, single-stranded or double-stranded, depending on the virus. The capsid is composed of protein subunits arranged in symmetrical patterns. Viral replication follows a sequence of steps begin with attachment, where viral surface proteins bind to specific receptors on the host cell membrane. After attachment, the virus enters the host cell through membrane fusion or endocytosis and releases its genetic material in a process called uncoating. Environmental microbiology studies microorganisms in natural ecosystem and their roles in maintaining ecological balance. Microbes are essential for nutrient cycling, decomposition of organic matters, and transforming of chemical elements in soil, water, and atmosphere. Microorganisms plays critical role in ecosystem functioning. They decompose dead organic material, releasing nutrients back into the environment. This decomposition process maintains soil fertility and supports plant growth. Biogeochemical cycles describe the movement of chemical elements through living organisms and the environment. Microorganisms play central roles in these cycles by transforming elements such as carbon, nitrogen, and sulfur between different chemical forms. The slide is designed for quick revision before the examination. It highlights the most important conceptual areas that students must revise repeatedly. Rapid revision helps reinforce memory, strengthen concepts links, and improve recall speed during the exam. Instead of rereading entire chapters, focus on high-yield topics allows efficient last-minute preparation. This slide identifies the specific topics that are most frequently tested in CUET PG microbiology, binary fission, antibiotic mechanisms, types of immunity, antibody classes, vaccine categories, nitrogen fixation, and viral replication steps are considered core conceptual areas. These topics represent fundamental biological processes that examiners often use to frame analytical questions. To master high-yield topics, focus on understanding mechanisms rather than memorizing lists. For example, know the sequence of viral replication, understand how antibodies recognize antigens, and explain nitrogen fixation supports plant growth. If you can explain these processes logically, you will be able to answer both direct and application-based questions confidently. This slide emphasizes the importance of structured and guided preparation for competitive exams. Microbiology contains interconnected topics, so systematic learning helps integrate concepts efficiently. Studying randomly may lead to fragmented understanding, whereas organized preparation ensures comparative coverage of the syllabus. The final slide serves as a closing message, encouraging continuous learning and Engagement. Mastering microbiology requires consistent study, repeated revision, and conceptual understanding. Regular exposure to explanations, problem-solving sessions, and decision and discussion of doubts helps deepen understanding. Subscribe BioBrainz for free lecture series.